Hello, so we're going to start with the protein synthesis study guide here. And the first question asks us about the central dogma of cell biology. Now, that's a concept that we have been talking about ever since like lecture 3, right? It's something that somewhat of a reoccurring theme that we have been discussing. And the central dogma begins with DNA. And so, as you know, there are genetic information that are encoded within the DNA, and these information are necessary for the production of proteins, for example. So DNA is going to be copied in the form of RNA, and that process, as you know, is called transcription. And that takes place in the nucleus. Once you make the RNA, you're actually going to process it a little bit before you send it out into the cytoplasm. And inside the cytoplasm, the ribosome is going to attach to the RNA and perform translation. Okay, that happens in the cytoplasm, either at the endoplasmic reticulum, the rough endoplasmic reticulum, or just in the cytoplasm where freely floating ribosomes are available, and that's gonna turn into the protein. And we're gonna use a copy of DNA that we've made, and we're gonna use those instructions to make proteins. So here we have a chart that compares DNA and RNA. And again, this is something that we have been doing quite a lot up until this point, of course, but it's good to refresh our memory. So for DNA, it is, of course, double-stranded, the double helix, whereas RNA would be single-stranded. Type of ribose sugar, right? Type of ribose sugar that we have would be deoxyribose. Deoxyribose for DNA, and ribose for RNA. Next, we have nitrogenous base. That's just the ACT or G for DNA. And for RNA, instead of having thymine, we are going to have uracil instead. So ACUG. We only learned one type of DNA. I'm just going to write DNA. But for RNA, if you watch the recordings, you would know there are rRNA, tRNA, as well as mRNA. And they all kind of do different things, which we will summarize in the next table. So let's start with rRNA. rRNA. The R stands for ribosomal RNA. And this stuff, along with proteins, they would form the ribosome. So it is a component of ribosome. Right? And it plays a role in linking the correct amino acids together. Right? So you physically create the peptide bond that would link the amino acid together, and that happens at the ribosome. So there is another type of RNA, the mRNA, the messenger messenger RNA, right? And this is actually the copy of the DNA, a copy of a gene, right? It contains codons, right? Codons are the triplet codes, right? Contains codons that encode for amino acid. Okay? And the codon tells you basically which amino acid to connect together. Right? So in addition to rRNA, mRNA, there is also the tRNA. T stands for transfer RNA. And this one is responsible for bringing the correct amino acid to ribosome. Okay. And at the bottom of the tRNA, right, there is something called anticodon. Contains anticodon that is complementary to codons. Right? So in order for the tRNA to bind to the ribosome, its anticodon must be complementary to the codons on mRNA. Okay? So that's that. List 3 steps of transcription. So we have initiation, elongation, and termination. Initiation, elongation, and termination. And basically, the initiation is when the RNA polymerase, that's the enzyme that carries out the transcription process, right? So RNA polymerase binds to the promoter, right? And promoter marks the beginning of a gene, right? So in case you don't remember that, I'm just gonna put a star here. Promoter marks the beginning of a gene. And you want it to be easily pulled apart. So for that reason, it is going to be rich in A and T. There's a lot of adenine and thymine because there are only 2 bonds that are holding the A and T together. It's much easier to pull apart when you have lots of A and T's compared to C and G's, right, which has 3 bonds holding them together. So the transcription process starts when the RNA polymerase binds to the promoter. It marks the beginning of a gene. And in elongation, that is when the RNA polymerase will unzip the DNA, and then it will use the template strand to build a copy of the coding strand. Okay, so in the recordings, I told you the difference between the two, right? The coding strand is the strand that actually contains the codons, which codes for the amino acids. The template strand is just there for the enzyme to use as a reference to build a copy of the coding strand. Okay, so this one doesn't actually have the code. It's just being used as a template. And finally, the process will stop when it gets to the terminator. So in the termination step, the RNA polymerase reached the terminator. Okay? So the terminator sequence marks the end of a gene. And that's when it knows to fall off. Okay. So probably a good idea to maybe draw a sketch here for you in case you are still a little bit unsure. This is the gene, okay, and it's double helix, right? Okay, double helix like this with A's, T's, and G's. Okay. And so here the beginning part that marks the beginning of a gene, that's the promoter. Right? So the RNA polymerase binds here, it will copy the whole thing until it gets to the terminator sequence. Francisca, do you have a question? I see that you have unmute your mic. Do you have a question?
No, I don't know why it has unmuted.
Sorry, I couldn't quite hear you. Oh, okay, all right, never mind. Okay, so there we go. So this whole thing is a gene, right? And gene contains instruction to make the protein. So now draw and label the various part of the pre-mRNA and the process RNA. Okay, so what happens is after transcription, right, we have what's called like a pre-mRNA. Pre-mRNA. We only copy the gene, right? This whole thing, that's the gene, right? Everything else that's before the promoter and everything that's after the terminator will not be copied. And we have our pre-mRNA. So it turns out the stuff that we copy, right? Not everything is useful, right? So we have some regions that are exon. This is called exon number 1. And then some regions are called intron. Intron number 1, exon number 2, intron number 2, exon 3, intron 3, and then it could, go on for quite some time. But the thing is to know is that exons are coding DNA. Okay, they are the ones that will get expressed in the protein. Right? So remember, exons are expressed, whereas the introns are non-coding. Okay, we don't really use them for anything. So what we do is we are going to do RNA processing, right? Which will help remove the introns because they don't code for anything anyways. You remove the introns and you will stitch together the exons. Okay, so in the end, I just have exon 1, exon 2, and exon 3 like that. Okay, we're gonna be removing the introns. We don't need them. Okay. Now remember, we are talking about DNA here, and same thing for RNA. There is directionality associated with it. So there's a 5' here and a 3'. And this thing, which by the way is still in the nucleus, it's a little bit unstable. If you take this and let it go out to the cytoplasm, it's probably going to get broken down very, very quickly. So to stabilize it, what we're going to do is we are actually going to add a 5' cap right here, a 5' cap. Okay, so this is called a 5' guanine cap. Okay, which prevents this end from being broken down. And then on the 3' end, we're going to add a bunch of adenine. Okay, so this here is called a poly A tail. The longer the tail, the longer it will last in the cytoplasm. Sometimes you want the RNA to stay longer in the cytoplasm so you can keep on making the protein. Other times you might only need to make the protein for a little bit and then you don't need it anymore. In those instances, you would want to have shorter poly A tail. Okay, but the function of the cap and the poly A tail, these things, they will help stabilize. Right? These will stabilize the messenger RNA. Then it's ready to go out into the cytoplasm. Okay? Does anyone have any question? All right. Just give me a second here. I forgot to set up the poll. But I guess we could do that later. All right. Here we have some tables to complete. Okay, we have the coding strand that's being given to us, and then we have to find the messenger RNA. Right? So remember, the coding strand is the strand that we want to copy, right? That's the strand that we want to make so that we could use it to make the protein. So our messenger RNA is going to contain the same information as the coding strand. So we're just going to copy it. And because we're dealing with RNA, there's no thymine. Instead, we have the uracil. So AUG, AUA, CGA, AAU, GAU, UAA. That's it. Okay. So that is the messenger RNA. Right? So the relationship between the coding strand on the DNA and the messenger RNA is that they are identical, except U replaces T. Right? The uracil will replace T. So there's no thymine in RNA. Okay. So just to refresh your memory a little bit, here we have the messenger RNA. Messenger RNA, 5' to 3'. And the first code is AUG, right? So AUG codes for an amino acid. Right? So we would have a tRNA, a transfer RNA. It's going to be coming along. This is tRNA, and it's going to be carrying a particular amino acid. In order for the tRNA to bind to the messenger RNA here, the anticodon, which is located at the bottom here, must be complementary to the codon. So if the codon is AUG, then the anticodon must be UAC. Okay? UAC like that. Okay. So the relationship between the mRNA and the anticodon is that they are complementary. So if you have AUG here, the anticodon on the tRNA must be UAC. Next one must be UAU, right? GCU, UUA, CUA, AUU. Right? You must use the base pairing rule to figure out the anticodon.
Excuse me, Vincent.
Go ahead.
Yeah, I just wondering. I'm confused about like a codon and anti.
Codons.
Yeah. Because
Yeah.
I watched the recording times,
Mhm.
didn't find like enough information for the anti-codon. I was
Sure.
wondering if you can
Yeah,
explain.
for
Yeah,
sure. Okay, no problem.
thank you.
Yep. Okay, so give me a second, guys. Let me just try to pull a slide here so I can explain. All right. Okay. So this is a tRNA, transfer RNA. And the transfer RNA is just an RNA that folds back on itself and it forms this clover leaf shape, okay? So one end of the tRNA is gonna have an amino acid, okay? And the amino acid that it carries is going to correspond to the codon that is on the mRNA. Okay, so the codon here is UUU, right? Now, there is a table that you have to look at, right? To figure out, to figure out which amino acid the codon codes for. Let me try to pull the table in here for you. Okay, so you see this table here, which is in your study guide, and I actually posted one as well for you to download on eCentennial, right? So we were looking at the codon UUU, and I explained how to use this table on the in the recordings if you need more help. But first codon is U, second codon is U. So this row intersect with this column right here, right? And then we try to look at the third codon, which is also a U. So UUU codes for the amino acid PHE, right? Phenylalanine, PHE. So when we go back to here, right, we see that, oh, the codon UUU actually does code for the phenylalanine. But how does the tRNA know that it's the right amino acid to bring to this situation? Well, at the bottom of the tRNA, there is the anticodon. And the anticodon must pair up with the codon, right? So what's complementary to UUU? Complementary to UUU is AAA, right? So if the anticodon and tRNA is AAA, it will dock perfectly onto the RNA, and then the amino acid would be brought to the ribosome. Okay? If you have an other tRNA right here, okay, and this one is like, let's say this is UUU, right? And again, that's the anticodon, right? So what would the codon be? That's complementary.
AAA.
That's right, it would be AAA, right?
AAA.
Yeah, and if we look up AAA on our codon chart, which is right here, okay, what does AAA code for? Well, A codes for LYS.
ASN. Oh, LY.
Yeah, see
A.
A,
Oh yeah,
and then
yeah. LYS. Yeah.
right, LYS, right, lysine, right? So in that case, this would be the amino acid. Okay, but UUU would not be able to bind to this UUU, right? In order for the tRNA to come in, it must be complementary with the codon. So let's take a closer look here, right? And everybody should take a look at this. We have two amino acids here, LEU and SER, right? Which one corresponds to the anticodon GAG? Okay, so let's take a look. What are we gonna look up on the codon table? You need to have the codon table for the quiz, right? Otherwise, you won't be able to do a lot of the questions. So if this is the anticodon, what would the codon be? What's
That would
called
be CUC.
CUC? That's correct. Does anyone have question why it is CUC? You should ask now if you are confused, because we're going to be doing a lot of these things.
Why is it CUC?
Okay. Do you understand the complementary rule? That G and C pairs together and A and U pair together?
Yeah.
Yep. Okay. So that's what we're doing. If you see a G, the complementary one is C. Complementary to A is U, complementary to G is C. Is that okay, Aisha?
Okay. Yes.
Okay. So you try another one for me, Aisha, please. Again, this is the anti-codon. What would the codon be for AGU? Aisha? What pairs with A?
Is it U?
And what pairs with G?
C?
Good. And finally, U pairs with?
A. Oh.
Yep,
A.
you got it. Okay. So that's the codon. And the reason we need the codon is so that we can look it up on the table here. Okay, you need to have this table either with you now or look it up on the phone. It's called a codon table. And we will look up the code. So for the CUC, right? Let's look up CUC. CU, so it's this block right here. Okay, so CUC codes for LEU. LEU, okay, leucine. So there is the LEU right here. And that means it's gonna bind over here. I'm gonna have the LEU, which again is corresponding to the codon. Okay, we never look up the anti-codon on the table. We always look up the codon. So similarly, if we look up UCA, take a look at UCA right here. UC, so is this block. UCA codes for SER, right? Serine. So this one's gonna go right here, SER. Okay. So let's take a look at this messenger RNA, right? The purple thing at the back, that would be the ribosome. We have the large subunit and the small subunit. And what we have over here is that we have a bunch of codes, right? So the tRNA will bring in the next amino acid, and it has to be complementary to the codon. If it's not complementary, it won't be able to bind, and it will just fall off, and then the next one would try. Okay, and it keeps on bringing in the correct amino acid, and then you keep on building the chain. So that's the relationship between the codon and the anti-codon. Is that a little bit better? Any follow-up questions, anyone at all?
Yeah, it's much better. Thank you.
Okay, thank you. Okay, so here we go, back to this table. Now we know the anti-codon. Now we have to look for the amino acid, right? Okay, so we have AUG. Let's take a look at how to look this thing up. Oh boy, there's a lot of stuff right. AUG. So first codon is right here, A, U. So you just use your fingers to trace it, and then you see where they intersect, right? So AUG, which codes for MET. Okay, methionine. You don't have to know what they stand for. I mean, it's just nicer to say it out, like what they code for, instead of just spelling it out. So this is MET. Okay. What about the next one? What would the next one be? We would look up AUA, right? So please take a look at the codon chart and look up AUA. What does it code for?
ILE or IIE?
That's L, yeah, you're right. ILE, that's absolutely correct. Okay, so AUC will code for ILE, okay, isoleucine. So that's what we're gonna put. I-L-E, A-U-A, sorry, mother. Do you guys understand how to use the chart? If you don't know how to use the chart, you need to let me know now, okay? So that I can explain to you one more time, right? Because if you don't know how to use the chart, you won't be able to do a lot of the questions on the test, quiz, both quiz and test. Does anyone have any question about how to use the chart or where to find the chart? If you can't find the chart, you should also let me know, and I'll tell you where to get it.
Vincent,
Mm-hmm.
the other time I told you that I cannot see the PowerPoint. I logged in with my phone. I've logged in with 2 phones, and I cannot see the PowerPoint slide. So I made someone downloaded it and sent it to me.
Good. Right. Again, right. I checked the file. I upload. Everything looks fine on my end, and I think other students are able to access it as well.
Okay, so Aisha is having the same problem. I don't know.
Yeah, I'm having the same problem. The PowerPoint slides are not coming.
What exactly is the problem? Can you elaborate a little bit?
You don't see the full thing. Okay, so if it's a picture, you see half of the head of the person. You don't see half.
Okay, so like I press on content, right? You go
No.
to the lecture, right? And then this is lecture 7. And then you are downloading the PowerPoint slide, right? This one, is that what you did?
Yeah, I try watching it, not necessarily downloading it.
Oh, sorry. Are you saying you cannot get the PowerPoint slides, or you cannot see the recordings? Because
No, I cannot see everything. You see,
just 1 second, Isato. Let me just mute you for a second there. So like the thing doesn't load up, right? Right, you're saying?
yeah,
So,
not
okay.
necessarily that, but you see, I'm having the genome of whatever here. So
Sure.
this one will be hidden, and I'll only see this part.
And then you don't see the rest of it. That's what you're telling me.
Yeah.
Okay. Now,
I see
did
half
you
of
try
it.
pressing the download button like I told you last
Yeah,
time?
I downloaded it and I saw half. I didn't see all of them.
Okay. What kind of computer are you using? Are you using PC or Mac?
Okay, this one is Lenovo, and I did that with my phone. I did that with an iPhone. It didn't work. I did that with an Android phone. It didn't work. So I don't know what the problem is.
Yeah. Okay. Anyone else having a problem here? Besides
No, we're good.
yeah, anyone else? I mean, I don't really. I'm not sure what the problem is. I cannot troubleshoot the actual computer problem. Maybe try to contact someone in IT. Maybe they'll be able to help you with that. But I know for certain, if you do this on a school computer, then it would guarantee to work. Okay, and I mean, you said your friend was able to send you the slide, right? So that's a workaround for that as well.
Okay.
And if all else fail, right, try to do it at like a public computer, like at a library or something. Yeah.
I asked Aisha, she's also having the same problem. So I told
Yeah,
it was my laptop. I did that with my phone. It didn't work. 2 phones, it didn't work.
Yeah. What about other courses? Are you able to get the stuff from other courses?
Yeah.
yeah.
I'm not sure what the problem is. I'm really sorry. But yeah, talk to IT and see if they're able to help you with that problem. And if it still persists, then just let me know. Then maybe I'll just email it to you guys individually. Okay.
Okay.
All right.
Okay, thank you.
Yeah, no problem. But back to what I was saying, right? Like, do you guys know how to use this table? Everybody knows how to use this table? Is that like?
I just have a question
Sure.
about like UAA and like UAG, how it says stop.
Yeah.
What does that mean? Is that just the word stop, or is that like still like an acronym or something?
Okay, great question. So these are the stop codon, right? And you know what? Let's hold on to that thought. Let's continue on here first, okay? So you know, the next one here, GCU. So we will look it up, right? G, C, and U. That would be ALA, right? ALA. Let me go back to here, ALA. And then the next one is UUA. So we go back here, UUA. That will be LEU. Oops. LEU. And then CUA. CUA. That would be another LEU. And then oh, sorry, I'm looking at the wrong one. I'm so sorry, guys. We should be looking at the RNA. Okay, we should be looking at the RNA. Let's try that one more time. Okay. It was CGA. You know, rather than switching back and forth, I'm just gonna put this thing right next to it. Oof, there we go. It's much easier to see that way. Okay, so one more time. Sorry about that. And we should be looking at the messenger RNA, the codon and the messenger RNA. So over here, CGA. So CGA, that would be ARG. Okay. We will look at the next one, AAU. AA. So right here, AAU. That is ASN. GAU. So GAU. Right, GAU. That's going to be ASP. And then we have UAA, which is stop, right? UAA right here, stop. So stop means is a stop codon. That's a signal that tells the ribosome this is the end of the line. You don't have to keep on making it, right? There are no more amino acids. You've finished making the whole protein. You should stop the process of translation. That's what it means. So there are 3 stop codon in total. We have the UAA, UAG, and UGA. And so those are the 3 stop codons that you should know. So it just means that you should stop the process of translation is all.
Sorry, I thought the GCU should be ALA.
GCU, which one is GCU? GCU?
Yeah, that.
Oh, this one, yeah, no, we're looking for the mRNA, okay? I made a mistake earlier. We should be looking at the, yeah, mRNA, not the anticodon, right? Anticodon is on the tRNA, right? Okay, so let's try another one here. We have the template strand. The template strand. The template strand is going to be used by the RNA polymerase to build the complementary strand, right, for the messenger RNA. So when you see TAC on the mRNA, on the template strand, the mRNA is going to be AUG. The relationship between the template strand and the mRNA is that they are complementary. Okay. If I give you the coding strand, it's easy. You just copy them and replace the T with a U. But if I give you the template strand, you would have to use the complementary base pairing rule. So GGG becomes CCC. CCT, what does CCT become?
GGA.
Sorry, one more time. CCT, what does it become again?
GGA.
Yep, you got it. GGA, right? Someone else for ATA. What does ATA become? I mean, I don't want to be calling out names, okay? But like, help convince me that you guys understand, so you will do well on the quiz. Type it in the chat box, okay? If you don't want to unmute, that's okay as well. Okay, what would be the code if I give you ATA on the template strand?
UAU.
UAU, yeah. And Aksa, I'm sorry if I pronounced your name wrong. You got that as well. UAU, very good. Okay. CGC would be GCG. and then T would be UAA. All right. So the anticodon, which is the one that is on the tRNA, will be complementary to the messenger RNA, right? So that's going to be, let me just switch the color here. It will be, UAC, GGG, CCU, AUA, GCG, AUU. Okay, I'm just going to give you a moment to check that and see if you understand, if you have any question. Right? Feel free to ask if you need clarification. Then we're gonna do the amino acid, right? And for amino acid, we always use the messenger RNA. Okay? Always use the codon in the messenger RNA. Right? So AUG, that's the start codon. Right? It marks the beginning of the translation process. And we can certainly come over here and see what AUG stands for, codes for. AUG, it's methionine, MET. It's always MET, the starting codon. Next, we have GGG. What does GGG code for? Francisco, you want to try that one? The GGG? Sorry, CCC rather, the CCC. What does the CCC code for?
Can I? Oh, sorry.
Yeah, go ahead.
Is it the PRO?
CCC. Right here. That's it. That's the PRO, proline. Very good. Next one we're going to look at is GGA. What is GGA? GGA.
GLY.
GGA. Let's see. GGA right here. GGA is GLY. That's good. That's good. Okay, so you see it's pretty straightforward, right? As long as you have the chart, have the table, right? You gotta have the table on the test. Right? I'm telling you. Next one is TYR, ALA, and then it's UAA. Right? UAA is stop codon, so we put stop. Okay, you should memorize the stop codon, right? In the PowerPoint recording, I was telling people to remember it, right? Like you got A in biology, you are awesome, you are good. Right? That's a way for me to memorize it anyways. So those are the stop codons. And anytime you see those things, you know to put the word stop. Okay. Okay, let's do another one here. This time I give you the messenger RNA and we're gonna have to work backwards to find a template strand. The relationship between the template strand and the messenger RNA is that they're complementary. So you use base pairing rule. Right? If you see an A, I know is gonna be a T. Right? We're in DNA again, right? So we can use T once again. Right? There's no uracil in DNA, so that's gonna be TAC. The next one is TGG, GTA, CGC, AAA, and ATC. Okay. Anticodon is complementary to messenger RNA. So we have the uracil now, right? For those of you who spot the correlation here, template strand and anticodons are actually the same, right? But the T will be replaced by the U. Okay. Any question? Okay, we're just gonna go ahead and do the amino acid. Let me pull this table a little bit lower. Okay, so UAG, that's methionine. ACC, ACC, it's gonna be threonine, THR. Next one, CAU. CAU right here is gonna be histidine. Okay, and then you just continue until you get to the stop codon, UAG, right? You are good, not stop. All right. I'm gonna give you a poll question here. Convince me that you understand what's going on here. Okay, convince me that I should not worry about the upcoming quiz, which by the way is Monday. So let's try this question, please. Okay, it's 1 minute. And I only have 4 answers. Okay. If you don't see the poll for whatever reason, you can still participate by typing the answer in the chat box, okay? Like, I just want to make sure that you guys know how to do this, right? Like, this is for you, right? Not for me, right? So it's important that you actually do try these questions. Okay, so let's share the results and see what happens here. This is the template strand, right? This is the template strand. And if I give you the template strand, then the RNA would be complementary to the template strand. Okay. So what's complementary to T? It will be A, right? And then what's complementary to another T? Also A. And then complementary to A would be U. So the first 3 nucleotide on the messenger RNA should be AAU. If you check these, C is the correct answer. AAU. Okay. A lot of you chose AAT, but we're talking about messenger RNA, right? There are no thymine on RNA, only uracil. Okay. Seriously, any question? Just ask whatever it is. Right? I'm sure if you have a question, other people in the class also have similar questions. So. Okay, no problem. If you change your mind later, feel free to give me an email. Okay, so that's that. Let's take a look at the next thing here. If a DNA sequence contains 360 nucleotides, how many amino acids do you expect to be in the protein sequence it encodes? Okay, so 360 nucleotides, 1 codon is equal to 3 nucleotides. Okay, which is 1 amino acid. Right? So if you have 360 nucleotides, you divide it by 3, that's going to give you 120 amino acids, assuming only exons are in the sequence. Okay, because we will remove the introns, right? They're the non-coding DNA. All right. I kind of missed a question here. Given the following mRNA sequence, which 2 codes for the same protein? So you would have to chop them up into triplets, right? And then you look it up in the codon chart, and then you compare them. Okay, see which 2 are the same. So you can try that on your own. I'll give you the answer. The first one and the last one, those are coding for the same amino acid sequence. They have the same primary structure, if you remember what that means. Okay, next we have mutations. I probably should give you a quick lesson on mutation. Judging from the interaction, maybe a lot of you haven't got that far in the recordings, if at all. Okay, so let's talk about mutation here. You should write this down. Okay, mutation. Mutation is a permanent change. Permanent change in DNA. So this could be caused by DNA damage. It could be caused by mistake that occurs during DNA replication. Could be caused by mutagens, carcinogens. All these things could change the DNA permanently, and we have a mutation. So we're going to look at 2 types of mutations. We're going to look at point mutation. A point mutation is a single change. Sorry, we can say a point mutation is a change in a single nucleotide. Okay, change in a single nucleotide. So there are 3 outcomes. Okay, there are 3 outcomes. Right? We can have, let's say, U. I know this is on the RNA, right? But the change actually happens in the DNA. It's just much easier for me to show you in the RNA so you don't get confused. Let's say we have U that will get mutated into UU. Let me make sure I pick the right one. UUC. Like that. Okay, so this is the normal one. We call that the wild type. And this is the mutant. Right, you see the 3rd nucleotide changes from being U to C, right? So in the DNA, it would have gone from a thymine, a T, to a cytosine. Someone please tell me, using the codon chart, what does UUU code for? Please look it up.
Is it AAA?
I am looking for the amino acid that it codes for. The amino acid that it codes for. So you have to use the codon chart to figure that out.
Is it PHE?
PHE, that's correct. Okay, so let's take the chart over here again, and you can see UUU codes for PHE, right? Very good. Okay.
So I'm sorry, do we have 2 different chats?
No, it's the same chart. Which other chart are you thinking of?
Oh, the one that has the UT something.
The UT something?
No, GC. Never mind.
Yeah, it's just the same chart, okay, that we always
Oh,
use. Okay.
oh,
Yeah, this should be the one that you'll be using on the test, okay? I'll post it on the announcement after class if you couldn't find it. Okay, what about UUC? What does UUC code for? Someone else. Let me just pick on someone here. Sherelle, can you tell me what UUC code for? Cheryl, is it Cheryl? I think it's Cheryl, right? Cheryl iPhone, it says here. I'm sorry if I mispronounced your name. Nope. Okay, maybe some of you are not here. Sarah, do you know the answer for this one? UUC, what does UUC code for?
Just give me 1 second. Would it be PHE?
Yes, PXG, right? Very good. Right. PXG. UUC also codes for PXG, right? So in essence, what happened, right? Did anything change? No, not really. The genetic code changed, but then the amino acid did not change, right? So this is what we call a neutral mutation. Neutral or silent mutation. Okay. Where it doesn't have any lasting consequences because it codes for the same amino acid. All right, so let's take a look at the next one, which we call missense mutation. Missense mutation. Okay, so this one would be like starting from UUU again, and this time I'm going to mutate it to UUA. So that's the wild type, and it's coding for PHE. What about UUA? Estelle, could you tell me what UUA codes for? If you're there, please. Okay, what about Fasama? Oh, you're there. Okay, you go ahead. Oh no, that's Francisca. Yeah, sure.
Is
Yeah.
it stop?
UUA? No, not quite. Not quite.
Oh
Okay,
my God.
UU is here, right? Okay, so you look at UU and then A, right? So that would be LEU. Okay,
Okay.
LEU. All right. So it changed. It become a different thing, right? So that's a mistake. It's a missense mutation. Okay. And then the next one goes like this. We have like UGU, and then it becomes say UGA. Right, so let's look up UGU. UGU codes for cysteine, CYS, and then UGA. It's one of the stop codon, isn't it? Right. So that's a nonsense mutation. Nonsense mutation. Okay. So what is the problem with this? Well, let's say you have a long chain, right? So you have a bunch of code, right? UC, C, CAC, CGC, UGU, dot, 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 very, very long. And eventually you have like a UAA, a stop codon. If there is a mutation that happens like in the middle, right? Like here, and it causes this to become an A, and then you would have amino acid, amino acid, amino acid, and then all of a sudden you would stop. Then the rest of the protein is not being made, right? So you end up missing a whole chunk of your protein, and the protein is probably not going to be functioning properly, right? So that's what we call a nonsense mutation, right? And it creates a truncated, a shortened protein. Okay, so if you must have a mutation, okay, what kind of mutation is the best one to have? Probably neutral, okay? That's the one that's not gonna affect you. Missense, I mean, it depends where it happens, and it depends what kind of amino acid is being replaced, right? Remember in lecture 2, we talk about how different amino acids have different properties, right? Some are acidic, some are basic, some are positively charged, some are negatively charged. So if you replace an acidic amino acid with another acidic amino acid, then maybe it's okay. Right? It's not going to have a drastic change in the protein function. But if the mutation caused an acidic one to be replaced by a basic amino acid, then that could drastically change the behavior of your protein. Right? So it depends on where it happens and how it happens. The outcome could be very different. Okay, so let's say for nonsense mutation, the stop codon, if it happens in the middle of your protein, then you lose a whole chunk of it. That could be pretty bad. But if the nonsense mutation happens later on in the sequence, right, then you end up not losing as much, and that probably is going to be okay. All right, Francisca, you you said you're confused. Do you have a specific question you want to ask so I can address it? Did you have a chance to watch the recordings before the class, or like in general, like remember this is not a substitute for the recordings, right? If this is the first time you're hearing me explain about these things, then you won't be able to get the full picture, right? That's why it's important to watch the recordings during the asynchronous portion of the class, the Monday class. And then
No,
come
I
in.
made someone sent me the slides, but
Mhm.
my problem now is how to use the chats. But because it looks like whatever I say is wrong.
Okay, sure. Let's do one together. Okay, so ACA. How do we look up ACA? Okay. A is right here. Okay, that's the first code. So this is the block that we're going to be looking at. Okay, C is right here, right? See that? Okay, so that means what you need is somewhere between the yellow and the green intersection. And we are looking for ACA, so A is right here. So that goes for THR. Okay. Now you try. Look up CUG. What is CUG? Go ahead. You there, Francisca? What is CUG?
L
Sorry, one more time, please. I didn't quite catch that.
LEU.
Yeah, right. CUG is LEU. You got it. Okay, I hope that's a little bit better. Okay, everyone else, are we okay with the mutation here? Point mutation. Any question about the point mutation before I move on to the next type of mutation? Okay. Number 2. Okay, we have the insertion or deletion. Okay, mutation. This is when you insert extra nucleotides or remove nucleotides from it. Fasama asks, are we going to have the chart on the test or quiz? You gotta print your own, right? Like, or have it on the phone when you take the quiz. Like, you're not gonna see the chart like right next to the question, right? Like, you have to have your own to use as reference. Okay. And I said this before, like I don't know if I'm like you know, hammering this point to you guys, even though like it's like a test, right, or quiz, whatever, right? Like, I am giving you permission to have your notes in front of you. You are encouraged to have a help sheet. Okay, I wouldn't recommend having the whole binder there because it would take too much time to look up things, right? And I wouldn't go in with your binders and not study. You're not going to do well. Okay, but if you have study, it's okay to have a sheet of reference information, right? Things that you can look up easily for things that you have trouble memorizing. Okay, so for example, it's good to write down what the stop codons are, maybe. Okay, just very quickly you can look that up if you need to, right? Maybe it's good to have a list of an enzyme there and what they do, right? Okay, it's okay to have that. Right? Actually, creating those sheets could be part of your studying a routine, right? And I encourage you to do that if you haven't been doing it. It's not cheating, okay? Because I create a test with that in mind, right? That you guys are gonna have access to this information, okay? So don't feel like you're, you know, cheating when you do that. For my course, okay? I can speak for other subjects. For my course, is okay. Okay, what is insertion and what is deletion, right? So let's say we have, and again, I'm just writing the RNA sequence because it's just easier for us to see. Okay, so that's the mRNA sequence, and I'm just gonna chop it up here into little triplets so we can see it easily. I'm gonna roll down our chart here one more time, and let's just translate this thing. Okay, so we have AUG. AUG is MET. Okay, and then the next one is CCA. CCA, that's gonna give me the pro. UUU is PHE, and AAA is gonna be LYS. Okay, so this one doesn't stop, but that's okay. It might go on a little bit, right? It doesn't really matter. We just try to illustrate something here. So that is the original sequence. Yeah, Ochiko, you have a question?
No, I don't.
Oh, okay. Because I just saw you unmute. That's okay, no worries. So this is the 4 amino acids. All right. Now let's see what happens if we have an insertion mutation. Okay. So I'm gonna have AUG, and then I'm gonna pop in an extra G or something. So you're gonna have GCCA. Everything else I just copy, eh? All right, so there is a extra G that is being inserted here. So now, what's gonna be the amino acid sequence? Well, first one is okay. First one is AUG, which is same as before. And then the next one, instead of CCA, we're gonna have GCC. Right? What does GCC code for? GCC codes for ALA. AUU codes for ILE. And then UAA, it's a stop codon. Right? So there's a couple problem here. First of all, this thing probably keeps on going, right? Because we haven't reached a stop codon yet. But now, because we have inserted an extra nucleotide, only the first amino acid is correct. Everything else after that is wrong. And not only are they wrong, we also introduced a stop codon. So the rest of the protein is not being made. Okay? Even if there is no stop codon here, even if it's able to keep on continue making, whatever amino acids, they would all be wrong anyways. So you wouldn't be making the original protein. You'll be making some random protein with unpredictable function, if there are any function associated with it at all. Do you understand? So when you insert something, and for that matter, if you delete something, you're gonna change the reading frame of your DNA sequence, of your codons. And that will result in extensive missense mutation, and sometimes you're going to introduce a nonsense mutation as well. Okay, so that insertion deletion mutation. The best kind of insertion or deletion mutation, well, I shouldn't say best, but then the least damaging, okay, the least damaging kind of insertion or deletion mutations are those that are multiples of 3. Right? Because if you insert 3 nucleotides, like if you insert like a CCG or something, then you would just end up having an extra amino acid in your sequence, and that might not be too bad, right? In terms of affecting the function of the proteins, having an extra amino acid might be okay. It's certainly much better than having a bunch of wrong amino acids in your sequence, and it's certainly better than missing a whole chunk of your protein, right? So I hope that makes sense. Now we're going to go back to our study guide right here. And this is just like a summary that shows you the different types of mutation here, right? Which is what I just explained to you. Okay, so you can see this is the wild type, the normal strand, and we have MET, ASN, PRO, THR, and then stop. So in base pair substitution over here, you can see there is a mutation. Instead of ACA, it has been mutated to ACU, right? ACU. So that's a single base substitution. And if you look up ACA and you look up ACU on the codon chart, they would be coding for the same thing. Both of them would be coding for THR. So in essence, there's no change in your amino acid sequence, And so what do we call this kind of mutation?
Silence.
That's right, that is the silent or neutral mutation. They didn't do anything. Okay. In fact, we probably have a lot of silent mutation in our cells that we're not aware of because there are no consequences associated with them, right? Next, instead of ACA, we have CCA. And we changed the first one, and now it codes for PRO instead of THR. So this would be a missense mutation. And again, I cannot comment whether it's good or bad. It really depends on where it's happening, right? The type of replacement we have, right? And other factors. It could be bad, right? Something like sickle cell anemia that's caused by a missense mutation, right? In amino acid number 6 of your hemoglobin. And that has very, very bad effect. Okay, next one here. In the second codon, instead of AAC, it got mutated to UAA. Okay, so they actually changed 2 of the nucleotides, and it became a stop codon. So this is a nonsense mutation, right? You will be missing an entire protein. Okay, that could be pretty bad. Okay, so insertion. Sorry, this is deletion. You took out the C, and after you take out the C, you miss the original stop because in the new reading frame, there's no more stop codon, right? So your cell would go beyond the normal stop point and then make a bunch of additional amino acid, add a bunch of additional amino acids onto your protein. Okay, so this is frame shift. Okay, and you're gonna have extensive as in like a lot of missense mutation. Sometimes addition could create a stop codon that wasn't there before. You see there's AAC before, but if you put a U right there, then the ribosome will read AUG and then it will read UAA, right? And then it will stop. So this creates a nonsense mutation. Okay, so there are different ways to cause a nonsense mutation. And over here, if you delete a triplet, then you lose an amino acid. Loss an amino acid in the final sequence. Is that bad? I think it's pretty bad in this case because the original protein only has 4 amino acids. If you lose one, then you lost like 25% of your protein, right? That's pretty bad. But some proteins are really long. Some proteins are like hundreds of amino acids. And if you lose one, and that happens to be like in some non-important location, then maybe it's okay. If it happens in the active site and it changes the shape of the active site, that could be a problem. Okay? Again, it's location dependent, whether it's good or bad. We cannot generalize and say, oh, nonsense mutation is always bad. Not when it happens at the end of the protein, because you're going to stop anyways. And losing a few amino acid might be okay. All right. So number 10 is difficult. Actually, I should erase all this stuff as well. Okay, number 10 is difficult because you have to create your own mutation. I think I did this in the PowerPoint recording, some examples of it anyways. But we can do it again here. Okay, we can do it again. The following is a template DNA of a section of a gene. Okay, so template DNA, which means I have to create the messenger RNA first. So I'm going to do it here on the side. The relationship between the template DNA and the messenger RNA, and you should know that by now because I've been repeating it the whole class, right? Is that they are complementary. So if you have TAC, then on the RNA you would have AUG. CAG, that will give you GUC, CUC, UUA. And again, let me know if I'm not making sense here, okay? It's really hard for me to tell because I cannot see your faces, and I can't tell if you fully get this or not. And then we have TAT, which is AUA, and finally ATG would become UAC. Okay, so I'm just gonna put commas here to separate the codons by triplets. Okay, so I'm gonna have to translate this thing, and it's gonna be MET. You're gonna look it up on the codon chart, right? The one that I've been showing you all class long. I just have it on my other screen, so I'm just gonna look it up there. VAL, LEU, UUA is another LEU, GAG is GLN, AAU again, ILE, and UAC is TYR. Okay, so that's that. So what are we going to do here? Well, we are going to create a base substitution that results in a neutral mutation. Right? So I'm actually going to look at the mRNA. You can pick any of them. Pick any of them, anyone that you like. Let's say we pick GUC. Okay, GUC. So we're going to pull up our codon chart again. Where's my codon chart? Codon chart is right here. Codon chart. And then we're going to do GUC. So G, U, and C, right? That's going to be valine. Now I'm going to try to change it so that it's going to give you another valine, right? Another valine here. So instead of GUC, what else gives you valine? Well, on top here we have GUU, right? GUU, that's also valine. Right? So if I mutate GUC into GUU, then there will be no change. And that's going to be a neutral mutation. Okay. But remember, this is the messenger RNA. So on the template DNA, right? What's complementary to U? Complementary to U is going to be A. So I'm going to change this last one to an A. Okay. That's that. Next one here, create a base substitution that results in a nonsense mutation. Okay, so nonsense mutation means we need to have, let me just copy all this stuff, okay, because it's the same sequence. Nonsense mutation means we have to have a stop codon, right, prematurely. So what are the stop codons? You remember the stop codon? Right. You got A, you are good, you are awesome. That's how you can remember them. And you basically need to create, let me just erase the thing over here. We basically have to find a spot that will allow us to create one of these stop codons. Okay, so what's a good location? Look at all these things. What is a good place to make a stop codon? Anybody want to try that? Okay, so let's take a look at the third one here. This is UUA. If I change the second one to a G, then I have UGA, which is one of my stop codon, isn't it? Right? So if the ribosome comes along, it will make the first one, second one, and then it will stop because it will see UGA. That's a stop codon, right? And then you won't make the rest of the protein. Okay, so that is a G, and on the DNA, it will be complementary to the G, which means it is going to be a C. Okay. Can you tell me another possible answer instead of creating a stop codon using this one? Is there another possible location for that to happen? Anybody? Do you guys understand my question? Right? We have to create a stop codon, right?
I don't understand the question.
Okay. Do you understand what I did here, or that's also confusing?
I don't mind if you
Explain again.
take
Yeah,
it
okay, sure,
again.
no problem. Okay. So the question asks us to create a nonsense mutation, right? A nonsense mutation is a mutation that creates a stop codon before it's supposed to stop. Okay, like when are you supposed to stop making the protein? You're supposed to stop making the protein at the very end, right? Okay, so over here, if I change this this sequence to U from UUA, which codes for LEU, to UGA, then I introduce a stop codon at this position. So when the amino acid, when the ribosome comes and read the mRNA code, it will put MET as the first amino acid, VAL, LEU, and then when it sees the next codon, UGA, then it will stop. It would not progress, it would not go on. So the rest of the protein is not being made. That's what's called a nonsense mutation, okay? And that happens because we replace the second nucleotide with G, creating the UGA, creating the stop codon. My question is there another codon here that we could change to create another nonsense mutation?
The UAC to UAA.
Yeah, right, that's good. Okay, like because the sequence hasn't stopped yet, that means it's probably going to keep on going. If you change the UAC to UAA, that would be a stop codon, right? Okay, and Shil just said CAG to UAG. Which one is that? Yeah, this one, right? If you change it to UAG, that also works, right? Because UAG would be stop codon, right? So I'm just crossed it out here. UAG, and then instead of UAC, you can change it to UAA or UAG, right? So all these are possible answers. Okay? Are we okay?
I am. Thank you.
All right, thank you. Creating an insertion mutation that results in nonsense mutation. So let's do that again. All right, so this is our sequence. And this time, instead of doing a base substitution, we're going to have to insert something. Okay, we're going to insert a nucleotide to make it nonsense. Okay, so where can we insert it? Okay. Anybody want to try?
We have to do what?
You have to insert an extra nucleotide, an extra letter to create a stop codon. Okay, I'll do it maybe. Okay, and you guys can see if it makes sense. So over here, right, your ribosome will read AUG, GUC, CUC, UAA, and then what if I put an extra U here, right? Then the next thing it reads would be UGA, which stands for codes for a stop codon, right? Then nothing else will be made after that. Okay. So that's how you do it, but we must reflect that in our original sequence because this is a template strand. So if you insert a U here, it means that we inserted A over there. Finally, create a base substitution that results in missense. That is very straightforward. Let's say we take the G here. Let me just copy that one more time. Oh, sorry. I'm just gonna erase this one then. Okay, let's say I take the G here and I change it to C. If we look up CUC on the sequence on the chart over here, CUC, it codes for LEU, right? Which is different, and that's a missense mutation, right? But if I change it to C here, that means I will have to change that to G in the DNA sequence. Okay. All right. Let's take a look at this one over here. Answer each of the following questions related to the mutation. So this is a template strand, and I give you the original DNA sequence. So what you have to do is you have to come up with a messenger RNA sequence here. Right? And again, it's best to chop them up into little triplets. Okay, and then you write the mRNA sequence. Okay, why don't I give you guys like a couple minutes to try this out on your own if you haven't already. All right, and then we will take it up. Let me just copy the chart for you. Okay, so go ahead. I want you to make the mRNA sequence, make the amino acid sequence, and then also do it for the first box right here. I'm gonna give you about 5 minutes to try it, and then we'll take it up. All right, go ahead, please. All right, I'm gonna try my luck again. Does anybody want to share their answer for the messenger RNA sequence right here? I know it takes some courage to do it, but anybody want to share it?
It's complementary, right?
Yep, very good. So
So I have AUG,
yes.
UGG,
Mm-hmm.
AAC,
Yep.
CGC,
Yes.
UGC,
Yeah.
and UGA.
You got it. Very good. Okay. I hope everyone else also got that as their answer. It's complementary because I gave you the template strand, right? If I gave you the coding strand, then you just change the T's to the U's. Okay, does anyone else want to give me the amino acid sequence? Amino acid sequence, please. That's all right. I will do it then. It is MET, TRP, ASN, ARG, CYS, and then stop. Okay, so that's the wild type, the normal, non-mutated version. Okay. What's the messenger RNA sequence for the mutated sequence here? Okay, so we can chop it up into triplets, just so that it's easier for us to see. Okay, and we'll check them one by one. Okay, TAC, same thing. ACC, ATT. Oh, there we found the problem, right? Okay. So everything is the same. The first one is the same. Blah blah blah, and then we would have, instead of having UGG, because of the change, we would have UAG, and then da 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 da. Right? Yeah, so what would be the amino acid sequence? Well, the first one didn't change, so it would still be MET. And then UAG is a stop codon, so nothing else will be made after that. Will there likely be an effect? Yeah, for sure, because you lost your entire protein, right? And this is a nonsense mutation. Okay. So you should try to go through these on your own, and I'll post the answer for you to look at. Okay, and that's pretty much it. The quiz, like I said, is going to be Monday. And then just hold on a second. I'm gonna see what happens next week. I think next week is the lab. Let me just double check that. You guys are the online one. Yep. So next week there will be the microscopy lab. We will not be having a synchronous class next week. You will work on your lab. You will do the quiz on Monday, and then you will submit it on Wednesday. Okay. Now, the lab is pretty straightforward. I'm gonna spend a little bit of time talking about it right now with you. Let me just open the file here. Okay, I'm just gonna go to my other class here. So I will post this online afterwards for you. So if you click on the parts of the microscope, you are supposed to read about it first, and do read it. Okay. Last time a lot of people didn't watch the video; they just tried to do the lab report, just based on what they knew or what they thought was the right answer, and that's why a lot of people got low marks. So you got to read through this stuff. Okay. There is a video that shows you how to use and care for the microscope, and then I will have the lab report for you to do, and then the lab report will have some calculations that are involved as well. And I will post a video that shows you how to do those calculations. Okay, so this is a video that I created that explains it. You should definitely watch it. It's only how long is it? It's only 15 minutes ish. It will clarify how the calculation needs to be done. So you're gonna have the right answer basically. Okay, so let me show you what the actual lab looks like. And of course, I'll post the link to it so that you don't have to look for it yourself. Okay, so because this is a virtual class, right? You will just be using a virtual microscope. I know it's not ideal, but it's the closest thing that we can come up with. If you're ever on campus, feel free to find me, and then I'll gladly show you how to use an actual microscope if you want. But this is a simulation. Okay, so you will be using this to do the lab, and the lab report is right here, microscopy lab. So it tells you what to do. That's the link to the virtual microscope, and then it tells you what to do. So click on learn, right? Click on learn right here. And then it has all these different parts, right? So if you click on it, it will tell you, oh, this is the body, this is the coarse focus, and then it gives you a description of what the function is. Okay, so that will help you label some of these parts, right? You try to match these letters to the names of the parts. Okay, and then it will tell you to click on the main menu again. Right? So main menu. And then you click on explore, right? You just follow whatever the instruction says, right? So there are some slides here that you're gonna look at, okay? And you will have to take a screenshot of what you see, okay? On the microscope. So like when you choose a slide, I'm gonna choose a sample slide, like the letter E or something, right? It's out of focus now, so you have to play around with the coarse focus and the fine focus, and then you just take a screenshot of this and then put it here. Okay, if you don't know how to do screenshot, you can let me know, and then I'll tell you how to do it. Okay, but you can probably search that up online as well. And then you will have to answer some questions. What is the relationship between the image, which is what you see, and the actual letter on the slide? Okay, so these are just observation stuff that you have to do. There are some calculations that you have to do here. And if you read the explanation, you will know how to do it. Okay, fairly straightforward. And then this is the part that you have to watch the video that I'm going to post. Okay, because it involves looking at a ruler and then figuring out the DFOV, the diameter of field of view, and that could be confusing. Okay, and a lot of people lose a lot of marks in this section. Okay, this is an older link. It will still show you like a sample calculation. But then the video that I post you, it's like a recent one that I make. It's a lot more detail, and that's the one that you should use. Okay, I don't know why there's a question mark here, but just ignore them. Okay, they don't mean anything. Okay. And yeah, that's basically it. Okay. At the end, you have to get more pictures for these phase, right? You have to find a cell that looks like interface. Don't copy like your whole screen. And ask me to find a cell. I just want the individual cell that is actually undergoing an interface, and you will paste it here. Okay. And there's some description here. This is very straightforward lab. Okay. It's certainly much easier than the first one, other than the calculations. Okay, but the calculations, once you watch the video, I'm pretty confident that you're gonna get it right. If you still have problem, sure, find me, no problem. It's gonna be due next Wednesday, at the end of next Wednesday. Okay, so no class, no synchronous class next week, and we will resume in March 27. Okay, March 27. Sorry, March 27 is the asynchronous class. We'll actually meet again on March 29 for a face-to-face, synchronous session. Okay, that's it for today. Thank you for coming, and enjoy the rest of your week. Okay, bye for now.